I hope you have a good lunch. And now we have a session, a special session, another, with three fantastic speakers. The first is Professor Vladimir Zarubaev, the head of the laboratory of St. Petersburg Pasteur Institute in Russia. Professor Vladimir searched and developed of novel antivirals among synthetic and natural compounds, studying of their targets and mechanisms of activity, development of animal models of viral infections to assess action of antiviral in vivo, studies of virus resistance to experimental drugs. It is a pleasure to have here Professor Vladimir. The talk is yours.
Thank you, Zilia. Thank you, dear colleagues, especially those who found forces to stop dancing. And of course, thank you very much to all organizing committee and Dr. Ana Paula personally for inviting me here. This is my first time in Brazil, and I'm praying that it would not appear the last one. I hope to come back one day. Thank you. The topic of my presentation is chemical modification of plant metabolites to confirm these metabolites' antiviral properties. First of all, I will try to answer the question, why do we need new antivirals? I will try to explain on the example of antivirals against influenza. The drugs of first generation were amantadine and rimantadine. They have been developed in the 60s. They have been introduced into wide clinical practice in the 70s. They were good, effective, and satisfied clinicians completely. But starting from mid-90s, physicians start to know that the efficacy of treatment began lower and lower, and scientific guys started to know that the rate of drug-resistant viruses started to increase. And currently, all influenza viruses are amantadine resistant because the mutation of amantadine resistance confer virus more strong, more forces. It can overcome the wild-type virus. The drugs for second generations were inhibitors of very important viral protein, viral neuraminidase. Now there are 4 of them, oseltamivir being the most popular. And when the physicians raised the question about the viral resistance, Hoffman and LaRoche said, no, 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 we promise, no resistance. This is a drug of a new generation, new drug, inhibitors of neuraminidase. However, in my lab, we started to know this since 2005, and all the labs in the world started to know this 2 years later. But nevertheless, now up to before pandemic of 2009, the influenza virus reached almost 100% resistance to oseltamivir. Just imagine that to discover, to develop, and to introduce oseltamivir into practice, these took about 10 to 15 years, and 100% resistance have been developed in 1.5 years. That's why development of new antivirals is always a race where humans always lose. In 2018, yes, and this was not exclusive situation for Russia. This was typical situation for the whole parts of the globe. In 2018-19, US FDA have been approved new antivirals. One of these did not pass through the preclinical trial, and another one, baloxavir, passed, and now it is sold in drogeries under the trade name Xofluza. But as quick as in 2020, first detection of drug-resistant strains have appeared. So 2 years and resistant viruses appear. That's why new antivirals of new targets, of new alternative mechanism of activity are highly required and necessary and are of great importance. Where can we find new structures for development of new antivirals? This is a structure of all antiviral, not only drugs, but all remedies of for control of viral infections. All these blue ones, these are vaccine, we do not consider them. And all the rest are chemical compounds. And only about 20%, this orange part, they are completely synthetic. All the rest are either completely biological, completely plant, or inspired by plant scaffolds, or somehow connected with plant scaffolds of secondary plant metabolites. Several words about viral terminology, just for all the further text would be understandable. Every compound is a poison, every compound is a drug. It depends on the dose. So this is presented how virus dies with increase of the concentration of compound. And this blue line is how cells die with the concentration of compound. The more distinct are these two values of 50% effective dose and 50% cytotoxic dose, the better the compound is. So it means that the compound is selective. It means that it prefers to kill virus instead of kill cells. Because the virus specificity is that it cannot live without cells. It's intercellular parasite, and it does not exist, it's not alive without live cells. So if we divide cytotoxicity to efficacy, we'll obtain a value which is called selectivity index, which should be 10 or more for prospective compounds. And another important thing is that viral cycle is a highly orchestrated process. When all the specific viral proteins work on specific time frames. There are initial phases of cycle, which is absorption to the cell membrane, penetration into cytoplasm, intermediate stages like transcription, translation, protein synthesis, and late stage of viral cycle, which include virion assembly and budding of progeny virions. We collaborate with many teams of chemists, both in Russia and abroad, and this consortium includes Vladimir University, where respective Professor Vladimir is from. And meanwhile, I invite all of you who is interested to join our friendly consortium and to take part in development of novel antivirals. We have a very wide viral collection, a representative panel of different viruses of different types and subtypes. We work most with influenza and enteroviruses, but it's possible, it does not depend which virus we work with. So I will give you several examples describing how the plant metabolites are or may become strong antivirals. The first example is dihydroquercetin. It's a flavonoid isolated from the larch. It's just a routine flavonoid, very low toxic with a very high antioxidant activity, but this is not why we love it. We love it because it decreases the level of reproduction of Coxsackie virus on animal model of viral pancreatitis. If we infect mice with this virus, their pancreas will be inflamed, will be destroyed, just routine pancreatitis. And if you see This is the level of viral reproduction in this organ. The classical antiviral ribavirin decreases it 2 orders of magnitude, and the hydroquercetin about 3 orders of magnitude, so it is much stronger than ribavirin, which is accepted to be active against any virus. And you see the application of the hydroquercetin highly improves the structure of pancreas. It highly decreases the number of infiltrating cells, the level of cell damage, tissue damage, and so the pancreas of infected mice treated with the hydroquercetin looks like almost normal pancreas. Second example is embelin, the alkaloid isolated from Asian plant Embele, that has been done in collaboration with Professor Veranut and Professor Buyart from Malaysia. First, they sent us a crude extract of Embele, and we demonstrated that it is active against the influenza virus. Then they isolated the major constituent of this extract, which is embelin. This has been published. And we demonstrated that embelin is more or less effective against different influenza strains. For some strains, it was more effective than reference compound rimantadine. For some of them, less effective. Meanwhile, against influenza B, rimantadine is completely ineffective, while embelin had more or less selectivity. So we showed that embelin is active on the early stage of viral cycle. It means that it inhibits viral absorption and penetration. And we knew that only 2 viral proteins are necessary at this part of viral cycle. The first is hemagglutinin, and the second is proton pump M2, so-called. We performed specific tests for anti-hemagglutinin activity. We showed that indeed embelin is able to decrease the activity of hemagglutinin, and this is how it interacts with the molecule of hemagglutinin. We modeled this by specific computer program. And second, and for my opinion, much more important example, is that chemical modifications can confer initially inactive plant metabolites strong antiviral activity. I will start with so-called cytizine. It's alkaloid isolated from golden radicacea. And as you can see, it is absolutely inactive. It has no selectivity. Its selectivity is too low. But our colleagues from UFA, from Chemical Institution, found a way to modify the initial structure of cytidine with different side groups. And I would like to attract your attention that this modification made some derivatives much more active than the initial inactive cytizine. We demonstrated that the most active derivative were able to inhibit the late instead of early stage of viral cycle, and we showed that its target is not viral but cellular. It changed the metabolism of cytoskeleton in such a way that cytoskeleton is not able anymore to support viral transport. So that's why this derivative has a very wide spectrum of activity, because all influenza viruses, maybe not only influenza, use cytoskeleton for transportation of their components to the cellular membrane. Example 2 is glycyrrhetinic acid. Only, I guess, very lazy scientists did not want to do something with this compound because it's very rich sources, it is used everywhere, it's like rice. You know, like rice. Well, this is its structure, and our colleagues from another chemical institute found a way to to make a substitution here and here. And they substituted it with either amino acids residues or nitrogen bases. And in fact, the situation was funny because it was natural compound modified with another natural compounds. And see how strong they become. So the selectivity of the last derivatives was more than 2000. It's very good. Yes, ginsenol, or ginsenol. Ginsenol is a cage alcohol found in the roots of ginseng. This is not an active compound as well. But our friends, our colleagues from a chemical institution have substituted this oxygen to this nitrogen. And this made possible to do very modest chemical library, but all these compounds became very effective against influenza H1N1 subtype. Some of them, if you compare with the reference compound oseltamivir, they appeared even more active, much more active, several times more active. It's very good. What is important that these compounds were effective not only in vitro, but in vivo too. It decreased the specific mortality of animals infected with a little dose of influenza virus. You see, it resulted in death of 80% of animals, and it decreased mortality very strongly. But unfortunately, when we started to investigate the spectrum of activity using different subtypes of influenza virus, we discovered that only those that belong to H1N1 subtypes appeared susceptible. All the rest appeared non-susceptible, appeared resistant to this drug, so there were no sense anymore to develop it further, because it's no sense to develop a compound against influenza which will be effective only against one subtype, as all the influenza epidemics are multi-etiology, H1, H3, H2, and influenza B. All the rest were non-susceptible to this drug. As for camphor derivatives, this example appeared much more successful. Similar to ginsamide, they took this camphor, this structure, with oxygen, and they substituted the oxygen to nitrogen, and based on this structure, they synthesized a very extended chemical library of about 500 or 700 compounds. Some of them I represent here, but these are not the most effective, and this one is most effective. We selected them from the whole library. We called it camphosin. It has low toxicity, high efficacy, and what is important for the manufacturing, very easy and low-cost synthesis. It has very wide range of activity against different types and subtypes of influenza. Its selectivity consists of about several hundreds. That's good. And it was active not only in vitro but in vivo experiments too. Here you can see how it decreases the specific mortality. What is important, it was active not only influenza A, but also against influenza B. And if you compare the activity of reference compound Tamiflu and camphosin, you can see that the activity of camphosin ever exceeded that of reference compound. It improves at a great extent the pathological structure of mouse lungs. It reduces the amount of infiltrating cells. It reduces the level of cell death. We found that it was active on the early stage of influenza virus life cycle. And indeed, it was active against specific activity of viral hemagglutinin. What about the resistance? Because the resistance to the drug is a very important, it's a headache, it's a nightmare of every virologist who develops the antivirals, because every virus is able to develop resistance to any antiviral. The answer was yes. The resistance can be easily achieved after 6 passage at increasing concentrations of campicin. You see the value of IC50 increased from 3 to almost 500. That means that to kill half of virus, now we need not 3 micromoles, but 500 micromoles, 160 times less susceptibility. That's bad. That's bad for potential antiviral. We found a mutation, we localized them, we calculated that indeed the molecular campicin binds with a much lower affinity with the mutated binding site. But finally, we decided to see what about the pathogenicity of this resistant virus. It did achieve the resistance, but whether it is of the same pathogenicity, is it still able to kill the mouse? You can see we infected several groups of mouse with equal doses of this virus. 10 after 3, 0 died in the control virus, which was campicin susceptible. 0 died in campicin-resistant virus. 10 up to 4, 30% mortality in campicin susceptible, 0 mortality in campicin resistance. 10 up to 5, all they died in the control viral group. Nobody died in campicin in group of mice which were infected with the same amount of the virus, but resistant. That means that it did become resistant, but it completely lost its pathogenicity. They even did not lose weight. You see, the blue lines are different dose of virus, of control virus, and red lines are campicin-resistant virus. They did not lose weight. And the level of reproduction in lung tissue was much lower as well. So the conclusion is that plant secondary metabolites represent almost an inexhaustible source of original chemical scaffolds. It can be modified in different ways, and it's endless search. And these compounds can demonstrate direct antiviral activity, and these scaffolds, which is more important, after rational chemical modification, can result in development of unique effective antivirals with novel targets and alternative mechanisms of activity. And my final conclusion is that only a rational and appropriate combination of virologists, botanists, phytochemists, and synthetic chemists would result in development of new antivirals and in success with the struggle with control of viral diseases. Thank you very much for attention, and a couple of more words. When chattering before the conference with my colleagues, I discovered nobody knows what is the weather like now in Russia. Nobody knows what is the golden autumn. And this is how Russia looks like now. This is a golden autumn, when all the leaves before defoliation become, instead of green, become yellow, red, orange, everything else but not green. Thank you so much.